It is my pleasure to introduce our work titled, Cell Type Specific Expression Analysis of Liver Transcriptomics with Clinical Parameters to Decipher the Cause of Intrahepatic Inflammation in Chronic Hepatitis B. This paper was published in the June 2024 issue of the journal iMeta. Worldwide, approximately 260 million people are chronically infected with hepatitis B virus, resulting in over 1 million deaths per year due to cirrhosis and hepatocellular carcinoma. The natural history of CHB can be divided into 4 phases according to HBe antigen status, HBV serum load, serum ALT level, and G-score. Results 1. As shown in Figure 1a, 82 CHB patients were characterized into 4 phases by E antigen status, HBV serum load, and ALT levels. PSEA demonstrated that patients with hepatitis could be distinguished from those with chronic infection based on their hepatic gene profiles in Figure 1b. The most upregulated genes in hepatitis phases were related to immune processes in Figure 1d. Apoptosis had the highest normalized enrichment scores among the known pathways of liver injury in Figure 1e. Result 2. As displayed in Figure 2a and b, 3 datasets of single-cell RNA sequencing data from human liver were merged and normalized. There were 13 main cell types were mapped using UMAP. The proportion of T cells was higher in livers of CHB patients in Figure 2c. Known marker genes were projected into the UMAP plot in Figure 2d. Hierarchical clustering used to identify intrahepatic cell type specific marker genes revealed 92 marker genes in Figure 2e. Result 3. Marker genes of immune cells were upregulated in liver samples in hepatitis phases in Figure 3a. Multiplex immune staining of liver biopsies showed that the fraction of lymphocytes was high in the infiltration site in Figure 3b. In Figure 4d, a multilinear equation that describes relative cell population sizes based on expression levels of marker genes. Figure 3e shows least square fits. An E-violin plot implied a slope distribution with a 75% confidence interval for each gene in the different clinical phases. Result 3. Marker genes of immune cells were upregulated in liver samples in hepatitis phases in Figure 3a. Multiplex immune staining of liver biopsies showed that the fraction of lymphocytes was high in the infiltration site in Figure 3b. In Figure 4d, a multilinear equation that describes relative cell population sizes based on expression levels of marker genes. Figure 3 shows least square fits. An E-violin plot implied a slope distribution with a 75% confidence interval for each gene in the different clinical phases. Result 5. We introduced a series of liver slides with CHB in 2 clinical groups, 12 patients with the antigen-positive hepatitis and 5 patients with the antigen-positive infection. Double immune staining of CTLA-4 and TIGIT with CD3, respectively, were performed on these slides. CTLA-4 and TIGIT were shown co-localized with CD3 at the inflammatory site from Figure 5a to d. We also conducted double labeling of CCL20 and CXCL8 with CD6, 8 and CD3, respectively. CCL20 and CXCL8 were widely expressed in CD6, 8 positive macrophages at the inflammatory site in Figure 5e and f. Result 6. There were 79 ISGs were upregulated in hepatitis patients in Figure 6a. We determined the beta values for these ISGs across different clinical phases in Figure 6b. Changes in beta values for these ISGs in different phases were shown in Figure 6c and 6d. The Bayesian model revealed a strong ALT-positive correlation with ISGs contributed by T cells and macrophages were involved in liver injury. Conversely, most ISGs expressed by B lymphocytes and hepatocytes showed no correlation with ALT levels. Hence, we inferred that these ISGs do not play a role in liver inflammation. Result 7. Double immune staining of IFI44 with IFI16 and ISG20 with ISG15 was performed on liver biopsies of 17 CHB patients from Figure 7a to d. In Figure 7e to g, IFI44 was proved to be highly co-localized with C antigen. Furthermore, the heat map displayed the areas of C antigen-positive cells in Figure 7h and indicated that IFI44 was highly expressed in C antigen-positive areas. Summary: this multi-omic study integrates liver bulk transcriptomic data, single-cell sequencing data, and clinical data to analyze the factors inducing hepatic inflammation in CHB, suggested that macrophages secrete chemokines to recruit immune-exhausted T cell. Innate immunity within hepatocytes is suppressed. Activation of ISGs in T cells and macrophages further exacerbates inflammation.